pleasure of hosting Dr. Andrea Quattrini, who joins us from the Smithsonian, and where her title is currently a research zoologist and curator of corals, which I think is really cool, and including all the great other anthrozoa, like anemones, and then also some fossil. She works also on fossil corals and things like that. But the coolest ones, in my opinion, are the octocorals and black corals of the deep ocean, which you guys are seeing in this rotating video here. So the general theme of her research program, I would sum it as, looking across ecosystems in the ocean, particularly around corals, because corals at any depth form the foundation of a keystone species in countless ecosystems, which provide homes and habitat for billions of organisms, especially in the deep ocean, which is really surprising to some people. Andrea has many publications spanning the gambit of understanding the ecological and evolutionary processes that shape patterns in marine biodiversity, genetic diversity, and phylogenomics, abiotic influences on species diversity, especially with depth and temperature and things like that in the ocean, seascape genomics, life history, biogeography, and even some fish stuff, which gets thumbs up from some of us nerds. And that, in fact, is how Andrea and I were fortunate enough to meet each other. We both are avid participants in the Okeanos Explorer program, which is a NOAA program to explore the deepest depths and farthest reaches of the unexplored deep ocean. And she has actually been a shipside and shore-based scientist. In fact, she's going to be what's the word for it, Andrea? I'm sorry. Oh, chief scientist. That's what it was. Yeah, she's going to be a chief scientist for upcoming expeditions. And she was fortunate enough, many accolades and awards for talks and papers, but also a recent Fulbright where she spent some time in Colombia pursuing her research interests. But without further ado, we here in the virtual space, we were unfortunate to not have her in person, but we're still happy that she was able to throw an audible at the last minute and join us in the virtual space. And I know you've been having a good day meeting with students and other faculty here at the university. And please join me in welcoming Dr. Andrea Quattrini.
Well, thank you so much, Randy. That was a really nice introduction. And thanks for the invitation. I'm really sorry that I'm not there in person. I was really looking forward to the visit. And also, I've never been to Michigan before, so I was looking forward to that trip as well. But it was really nice. I met a lot of students today and postdocs and had and faculty, and had a really great day. So I guess it's nice that we've moved to this virtual world, that we can do this via Zoom sometimes. So yeah, today I am going to talk about my efforts with collaborators and students, colleagues all around the world to rebuild the coral tree of life through ocean exploration activities, the use of museum specimens, and genomic discovery. All right, so corals are arguably some of the most important animals in the marine environment and perhaps on Earth. By their ability to engineer entire reef-based ecosystems, corals host tens of thousands of other species, supply numerous ecosystem services, and they provide sustenance to millions, if not billions, of people worldwide. I have no doubt that most of you are familiar with these ecosystems and the biodiversity that they support, but what if I told you that we actually don't have accurate estimates of their diversity? We don't know the full extent of their distributions, and we don't have a complete picture of their evolutionary history. Now, in part, this is because of several long-standing issues in coral systematics. And today I'm just going to bring up a couple of these points. The first really regards morphology and the morphological characters that have been used to discern one species from another and classify species taxonomies. Many of the characters that we use are plastic. In other words, they can change due to changing environmental conditions. This image here on the right is work done by David Paz Garcia, where he followed the change in morphology of a coral colony through time and noted that a species can change its appearance into another species. And this was just due to the amount of storm surge that occurred in the area. A lot of the morphological characters that have also been used are actually not homologous, and this has caused some distant related species to be grouped together inaccurately based on these convergent traits. In addition, corals and their sea anemone relatives have really slow rates of evolution in their mitochondrial genomes. And this has caused the use of DNA barcodes, conventionally used ones like CO1, for example, to really not be useful for discerning species within corals. Folks that have built trees with these barcodes these trees have been really poorly supported, several polytomies, and poorly supported across the entire tree from deep to shallow time. So these long-standing issues have really led to a poor understanding of the phylogenetic history of corals and their close relatives, the sea anemones. But in addition to these issues, there's really just undersampled diversity, even on these warm, shallow tropical reefs, as you see here. Some species that are thought to be widespread turn out to be cryptic, and there's multiple species. And beyond the sunlit warm waters of the tropics, there's corals that occur in the deep sea. Yep, they can be found hundreds of meters below the surface of the ocean and often hundreds of miles away from any shoreline. In fact, it's estimated that corals in the deep sea, at least greater than 50 meters, are more diverse than those found in shallow waters. This picture is of one species in particular that creates coral reefs in the deep sea. Now, there's just a few species that create coral reefs in the deep sea, and the majority of hard or stony corals in the deep sea are not, in fact, reef builders, but they're small and conspicuous, solitary, and often difficult to collect. The picture in the middle is of a cup coral that's just 1 millimeter in width. And the picture on the bottom left is a coral that we collected on a recent cruise off of Puerto Rico this past April. It's just a couple millimeters in width. And in addition to these stony corals, there's other groups of species that have really few active taxonomists working in these groups, including octocorals, which are also sea fans and soft corals, and the black corals. And all of these, as Randy said in the beginning, are really foundation species in the deep sea, also in shallow waters as well, and they support a diversity of associated fauna. I wanted to show you this chart. It's actually the new species descriptions that have occurred within the class Anthozoa, which is comprised of corals and sea anemones. The new species descriptions since 1758. And despite all the challenges that I've mentioned, there's been more than 200 species described in the past 5 years. This increase in new species is due to several factors. One, the feasibility in decreasing costs in genomic approaches, new analytical techniques in integrating different types of data to delimit species. I also think there's been more and improved targeted collections in deeper waters through the use of tech diving and submergence vehicles like submersibles and remotely operated vehicles. And in general, I think there's just been an improved understanding of the longstanding issues that we faced in coral systematics. And this has led to an increase in descriptions, primarily of cryptic species across all depths. And it's really a critical time to be improving coral diversity estimates, and critical time to documenting their full distribution and understanding their evolutionary history. As I'm sure you all know that, corals are really highly vulnerable to climate change. This is an image of a reef made of the genus Acropora, and this is a bleached reef. Just in brief, bleaching occurs when water's warm, corals stress, and they expel their symbiotic algae living inside their tissues. Over the past decade, we've seen several mass mortality events due to these warming temperatures. Now, for deep sea corals are also not immune to climate change. They are susceptible to ocean warming, ocean acidification, and deoxygenation. But I think of more imminent concern for deep water corals are activities associated with resource extraction, including fishing, for example. The image on the top is a trawl fishery off of New Zealand. collecting orange roughy. And the picture on the bottom is a bubblegum coral, just a portion of the coral colony that was collected as bycatch in this fishing, in this trawl industry. Activities associated with oil and gas extraction are creating impacts to deep water corals. And then the subsequent impacts like Deepwater Horizon or like oil spills and the Deepwater Horizon oil spill in 2010 that occurred in the Gulf of Mexico really occurred at depth at about 1,800 meters in depth. And we did see negative impacts to coral communities in that area. And of more emerging concern is mining minerals and precious metals from the deep sea floor. There are proposals to mine for precious metals like nickel and cobalt and copper from areas which we really don't even know what's there. But a lot of the areas that they're targeting are hard grounds and likely contain diverse coral communities. So I think to successfully mitigate all the impacts of global climate change and other anthropogenic stressors, really requires fundamental knowledge on how and where biodiversity is generated, how many species there are, how these species are distributed and connected, because we really can't protect what we don't know. And we need to better understand how the environment shapes that diversity across multiple scales of space and time, because we really can't save what we don't understand. And I would argue that at the core of this fundamental knowledge is taxonomy and an evolutionary framework, otherwise known as systematics, so that we can better document the extent of coral diversity and not just taxonomic diversity, but genetic and functional morphological diversity. We can better understand the contemporary and historical drivers of that diversity so we can inform the likelihood of persistence into the future. And so today I'm going to discuss 3 projects in which collaborators, students, and I have been working on to start, rebuilding this coral and sea anemone tree of life. The first project is going to be about the backbone phylogeny, which includes all orders of corals and sea anemones, and including some ancestral character trait mapping through deep time. The second projects that I'm going to be focusing on today is species delimitation into coral genera, including the incorporation of museum types into those efforts. And finally, because I really do think it's very important that we increase knowledge of the deep sea and continue to characterize biodiversity, I just wanted to share with you a little bit about ocean exploration and a recent expedition that I led off of Puerto Rico. Okay, so to begin rebuilding this framework, I developed a novel genomic approach, target capture enrichment genomics. It's not novel to the field, but it was novel for my system that I work on a couple years ago. And just in brief, I designed baits to capture or hook on, if you will, conserved regions of the genome. And then by doing this, you can also grab flanking regions that are more variable, more phylogenetically informative. So we capture about a couple thousand of these regions of interest in the lab with some molecular lab work, sequence them on a high throughput sequencer, conduct some bioinformatics and build trees. It's simple enough. And to date, since this original paper came out in 2018, we've published collectively about 11 papers, with many more currently underway, just really making considerable advances to the field of coral systematics. So to date, we published a few iterations of this bait set. I designed the first bait set to target around 1,791 loci in both corals and sea anemones, so across the entire class Anthozoa. Got the data back, and then we're able to redesign these bait sets to target octocorals, which are sea fans and soft corals, and hexacorals, stony corals, black corals, and sea anemones. And this is going to make a little bit more sense on the next slide. But basically here I added more loci and more baits to work a little bit better within these 2 different subclasses of Anthozoa. So I'm going to get right into the results here and show you the first phylogeny that we produced from this work. Really, again, to begin rebuilding the coral tree of life here. And there's tons of different stories to tell, but I'm just going to share a few highlights from this work. First, this is a well-supported tree. It includes two subclasses. Again, these are reciprocally monophyletic, the hexacorals and the octocorals. And this tree includes all groups of anthozoans, all orders, which include the Sclerotinia or the stony corals, the black corals, a few different orders of sea anemones, and two major orders of sea fans and soft corals here. So now that we had this pretty well supported phylogeny, I used just a few fossil calibration points to date this tree and found that Anthozoa originated before the Cambrian explosion. They've been around for approximately 770 million years. I then turned to mapping some different traits using ancestral state reconstruction onto the tree. And I focused on what are really like the hallmarks of reef building corals. So the ability to create a skeleton, a calcium carbonate skeleton, and thus engineer the structure of a reef. Coloniality, so being colonial instead of solitary, and then at least for reefs in shallow water, having the photosymbioses that are so important to shallow water corals. So I just want to take another second here to focus on the calcium carbonate skeletons. Anthozoans have a diversity of skeletal traits. So there's actually two different polymorphs of calcium carbonate that corals can create. And this is just basically different crystalline structures of calcium carbonate. There's aragonite, which is found in all reef building corals, and then calcite, which is found in the majority of the soft corals and the sea fans, excuse me, that have central skeletal axes. There's also some corals that are primarily proteinaceous. They don't calcify at all. And then sea anemones, of course, have no skeletal axes. And there are also several soft coral species that don't have a central axes made out of protein or calcite, but they do have small elements known as sclerites that are made of calcite. In fact, all octocorals do have these sclerites, these small skeletal elements or scale-like structures that are made from calcite. So I then wanted to just take a moment to focus here on the ancestral state reconstruction of these different types of skeletons. I'm not going to show you the proteinaceous skeletons today. I just kind of wanted to focus on calcite and aragonite. And I'm going to start here by saying that the most recent common ancestor of the Anthozoa were solitary. They didn't have a skeleton and they didn't have photosymbionts. And then looking at the first appearances of these other hallmark traits, we see that coloniality has been around for a really long time. Before the Cambrian, more than 570 million years ago. And we see within the hexacorals a few other appearances of coloniality and subsequent losses of coloniality in some of these groups, particularly the stony corals. The oldest origin of photosymbionts was around 380 million years ago. And this actually, this origin is with the Scleractinians, the stony corals. There were several other gains of photosymbionts closer to the tips of the tree, but this is the first appearance. And when we saw this at first, this kind of pushes the date back of photosymbionts back 100 million years, which at first seemed kind of impossible, but there is indirect morphological and isotopic evidence that some of the extinct anthozoans, they're both rugosins and tabulates that occurred during the Devonian and Silurian periods, that they likely had photosymbionts as well. And this corresponds pretty nicely to this date that we have. Okay, and then when do we see calcite? Calcite arises around 440 to 500 million years ago, and aragonite around 350 million years ago. And all, again, Scleractinians and stony corals build aragonitic skeletons, which seems to be a key innovation for the Scleractinia. And, you know, notably, throughout geological time, the oceans fluctuated between calcite and aragonite sea states. So for more than 500 million years, there were times when seas favored precipitation of calcite. There were times when seas favored precipitation of aragonite. And so we wanted to ask whether these past ocean chemistry conditions were coupled with the evolution of these distinct polymorphs of calcium carbonate. In other words, does the origin of calcite aragonite occur when seas favored those conditions? And in addition to this fluctuating chemistry, there were changes in temperature and carbon dioxide, of course, throughout several 100 million years. And deep in time, there was a high temperature, high CO2 world. During this time, it was difficult to calcify at all, particularly with high CO2. So mapping, again, these different calcite and aragonite states onto the tree, we can see a pretty good coupling of the origins of these different polymorphs with the past sea states. The origin of calcite occurred during calcite seas, and the origin of aragonite occurred during aragonitic seas. And I just want to mention again that the ancestor that didn't have a skeleton wasn't colonial, lived in a time of high temperature, high CO2, a time when it was really difficult to calcify. So just to summarize this part, we were able to use target capture genomics to really reconstruct a well-supported backbone phylogeny of the class Anthozoa. We were able to use ancestral state reconstruction to look at the first appearances of these really important traits of reef building corals. We generated general divergence times for many major groups and determined that anthozoans do have Precambrian origins. And, there were drastic changes in environmental conditions throughout the past several 100 million years. And corals and their relatives, the sea anemones, definitely experienced these drastic changes. But, high temperatures and high CO2 in the past were not conducive to reef formation, and will more likely, not be conducive to reef formation in the future either. So, now what we're doing, we've built this backbone phylogeny, is we're really continuing to build this taxonomic framework, adding more tips to the tree, adding museum types to the tree, revising taxonomy, delimiting species across all of the class Anthozoa. And when I say adding types to the tree, when a new species is described, you designate a type specimen, which is from a certain location, and you designate that in the literature as being the type for that species. And so we're really trying to integrate where we can types into these analyses. I'm not doing this by myself. This is a lot of work. Collaborators in Australia at the Queensland Museum and James Cook University, collaborators at the University of Sao Paulo in Brazil, and students are working on the stony corals. I have a postdoc who just started working on the black corals, started at the Natural History Museum. Collaborators at Ohio State and American Museum of Natural History are diving into the sea anemones. And myself, primarily with my postdoc advisor, Kathy McFadden from Harvey Mudd College, along with students and other collaborators, are digging into the octocoral group. And so today I wanted to focus on two different coral genera, Acropora and Paramerissia. Both of these genera are targets of active restoration and conservation in natural habitats. Acropora includes species that are extremely fast growing compared to other stony corals. And so it's being grown in nurseries to outplant on reefs. And Paramerissia was a genus that was impacted by the Deepwater Horizon oil spill in 2010. The image on the right is a Paramerissia colony, and you can see all this like brown flocculent material that was on it. It was subsequently, that material was fingerprinted to have that oil from that spill in that flocculent material. So we confirmed that Deepwater Horizon oil spill impacted these communities at depth. And so this species, this genus now is a real focus of current restoration plans that are happening in the Gulf of Mexico from the Horizon oil spill. So, I really think that for restoration programs to be successful, we need improved information, including what species should be restored and where do these species occur, To what degree are their populations connected? And again, how does the environment impact their distribution? We really don't want to outplant these species into some areas and then maybe introduce them to an area where they don't naturally occur, or introduce them into an environment where they're not going to survive. So I think at the core of this is accurate knowledge of species identities and their distributions, and thus coral phylogenetics. So Acropora is also one of the most species-rich and numerically abundant coral genera on most reefs throughout the Indo-Pacific. They're extremely diverse morphologically, and this is illustrated by the fact that over time, since 1815, more than 400 nominal species have been described. However, almost 3/4 of these nominal species in the genus have been considered to be synonyms or unresolved taxonomic revisions. And so there's only 122 valid species out of these 400 named species. And this is really due to assumptions of widespread distributions and some assumptions that the phenotypic variation that is seen is due to plasticity, which I talked a little bit about earlier in my talk. So I've been working with colleagues in Australia. They have been going out to several reefs all throughout the Indo-Pacific, collecting what we call topotypes, so representatives of species that were collected in the type localities. And they're really trying to do a solid effort at collecting all nominal species. Where we can, we're integrating museum samples into this work. For corals, for stony corals at least, most of the museum specimens include corals that are dried skeletons, and oftentimes they've been bleached, so there's no tissue left. But my postdoc, Michael Connolly, has been diving into our collections at the Smithsonian and realizing that there's several specimens that actually still have dried tissue. We're getting DNA out of that tissue. We have yet to determine whether there's any coral DNA in that tissue, but I'm pretty confident that we'll be able to use target capture enrichment of some of those types to get some loci out of those type material. All right, so we're using target capture enrichment of this tree includes about 2,425 loci, and to date we have only 154 individuals in this clade. My colleagues have been ramping up sequencing many more individuals. We just have yet to integrate it into this tree. And I just want to say, here's some of the locations where they're collecting all throughout the Indo-Pacific. And I'm just going to focus on the Acropora tenuis complex. This is the complex and the work that I'm most intimately involved in. And so zooming in on this complex, we were able to call SNPs from the target capture data and use various programs to look at structure and try to delimit species. So we've used SNAP and Bayes factor delimitation, structure analyses, unsupervised machine learning, and also very important to integrate morphological characters as well into this analysis. So in this clade, in Acropora tenuis, there are 11, what would appear to be 11 species. That's the preferred model. And 4 of these match nominal names. Excuse me. We definitely have 2 new species, and the remaining are likely new species, but really weren't further investigation. Some of you, if you can look across this, you might be wondering if some of these results really just reflect population structure and not true species boundaries. And this is a hard, answer to get to without further demographic models. And so some things that we are working on is incorporating models with gene flow into these analyses. But I do want to mention that some of these species were collected from the same locations, and they do occur sympatrically. So for example, Acropora tenuis was found with Acropora species SPP, which doesn't have a name yet. So again, we have a little bit more work to do here, incorporating different types of species delimitation models into these analyses. And I also just want to mention that there is an Acropora tenuis genome, and we were able to bioinformatically call these ultra-conserved elements and exons, or these loci that we're using to build these trees. And what we're finding is that the genomes aren't identified correctly. And they're not deposited in any museum. And so I really urge any of you who are working on genomes to really think about depositing your specimens, your voucher specimens, in a museum so other researchers can validate those IDs. Okay, so I want to take a moment now to talk about a genus Paramaricia that's near and dear to my heart. I started working on this genus during my PhD work in the Gulf of Mexico. Paramaricia is a widespread species, occurs throughout the North Atlantic Ocean, the Indian Ocean, the Pacific Ocean, and it's found in submarine canyon habitats, along seamounts, various different types of deep sea habitats, and complex topography in the deep sea. There are a couple species that occur in shallow waters, but most of the diversity is found greater than 200 meters at depths down to 3,000 meters. And, when I started working on this during my PhD, before knowing all the issues with some of the DNA barcodes, was able to generate a tree based on a couple genes, mostly mitochondrial genes. And, notice that there's a strong incongruence between morphological characters and these barcodes. So all of the tips here, all the circles are color coded to match a morphological species. And, if you look across this tree, you'll see absolutely no pattern. There are some species like the one represented in blue, Paramaricia biscaya, that has multiple barcodes, and they're found in multiple clades across this tree. In other cases, you might have the same species with different barcodes and different species with the same barcodes. And this was all just quite confusing to me during this time. And so I kind of came with the question, are these different species that just has variation in morphology, or the molecular barcodes just not informative enough to the limit species? And so I set out to answer this, and in this case, using RAD sequencing on individuals that we collected from deep sea cruises throughout the North Atlantic. So from the Gulf of Mexico up the US Eastern Seaboard to the Labrador Sea off of Canada, and then off of Ireland and in the Mediterranean as well. And I used restriction site associated DNA genomic sequencing for this approach, which is similar, in general to target capture genomics, but the first step is incorporating restriction enzymes to shear your DNA. So this approach is, for any of you that work on these different genomic methods, RAD sequencing has a lot of utility, and it's really nice, and it works really well when you have high molecular weight DNA, generally when it's preserved for genomics purposes. But it doesn't work so well if your DNA is super degraded, which of course most museum samples, unless they were preserved for genomics, the DNA is quite degraded. And in a lot of cases, even when we collect fresh tissue from deep sea animals, we still have a lot of degradation that occurs. And I think this is just due to them, depending on the depth that they're brought up, they can rapidly disintegrate, and their DNA won't be as high quality. So anyways, for this project, I use RAD sequencing, but I'm now going back in and doing target capture genomics to try to incorporate some of that type material that I've been talking about a couple times today. All right, so I was able to build a phylogeny using a genomic data set of around 250,000 base pairs and 2,800 loci. And just to tell you a few things about this tree, first, there's shallow mesophotic species which are early diverging, and at least for Paramyrecia macrospina, sister to a primarily deep water clade that's species found in greater than 200 meters depth. Again, using some various species delimitation approaches, we find that there's 7 species, and several of these are geographically widespread. So example, Paramyrecia biscaya, which is seen on the right, it's species number 7, is found in Ireland, which is denoted in green, and the Gulf of Mexico, which is denoted in blue. Paramyrecia plecomis, denoted as species number 3, is found in the Northeast United States and the Gulf of Mexico and off of Ireland. So in many cases, you have these species that are quite geographically widespread. Now, looking at the barcode data, this line here just kind of designates different what we call molecular operational taxonomic units just based on the barcode data alone. And the different colors correspond to the same MOTU, again, based on this barcode data alone. And you can really see there's no congruence with the RAD sequencing data. And I've just concluded that these conventionally used barcodes are just not informative enough to delimit species. And really, this is likely due to incomplete lineage sorting. What's interesting is looking back at some of the morphological characters, we can really find diagnostic characters that are fairly congruent with the RAD sequencing data. I've been able to put a couple names on these species, but again, we really have to go back and incorporate some of the type material, and using the target capture genomics, I'll be able to do that. And we need to look at the type material for the morphological characters as well. These 3 images that you see are scanning electron microscopy images, and you can see their close-ups of all the different sclerites or those calcite elements that I was talking about here. And those are diagnostic, often at the species level. Now, what's interesting, when we map temperature and depth of collection onto this phylogeny, we really see a strong pattern of phylogeny with depth and temperature. In general, we see diversification looks to that it proceeds from warm to cold and shallow to deep environments. So they're geographically widespread, but they're diversifying across a bathymetric gradient. And this has brought us to the question of what genes are under adaptive evolution along this depth gradient. You know, the deep sea can be a pretty challenging place to live. There's cold temperatures, there's high hydrostatic pressure, it's often food poor, and these pose, you know, physiological challenges and challenges to even maintaining a cellular integrity for these animals that live in this environment. So I really wanted to set out to address this question using a target capture approach. And I chose this approach over others, you know, instead of sequencing a genome and conducting perhaps some genome resequencing. I wanted to try this other approach because I work in a museum and I really want to be able to utilize the specimens that we have in hand. And can we utilize target capture genomics to target capture genes of interest, you know, whatever genes we might be interested in, and then capitalize on specimens that are housed in museums. So last year, my postdoc Danielle DeLeo, myself, along with 2 virtual interns during the pandemic, began this project. And my interns made this amazing video. They presented this work at a conference last year. And I thought instead of me telling you about this project, I would let them tell you. And I'm just going to play this video for you because I think they did a fantastic job.
Hello, my name is Sriram Ramamurthy.
And my name is Ariane Buchenmeyer. We're going to talk about creating a target enrichment bait set for deep sea octocorals to study how they respond to environmental change.
Metazoans, or corals and relatives, are ecologically important members of deep water ecosystems. However, most genomic studies on this group have focused on stony reef-building corals. Most octocorals do not have hard skeletons and do not build reefs, yet still are important foundation species in the deep sea.
Challenging environmental conditions of the deep sea may put selective pressures on deep sea organisms, which may lead to adaptation and speciation. Studying these evolutionary processes in the deep sea may give us insight into the factors driving biodiversity.
Target enrichment hybrid capture is an emerging method to conduct genomic and phylogenomic studies. Target capture is more efficient and cost-effective compared to other genomic sequencing methods and can even be used with DNA from historical museum specimens.
Our target bait set focuses on genes expressed across 3 octocoral taxa, selecting genes from both a previous study on an octocoral response to the 2010 Deepwater Horizon oil spill and a blast-based analysis from an exploratory literature review. Our interest is targeting not only octocoral or anthozoan-specific stress responses, but also exploring survival adaptations found in other deepwater organisms.
Our final bait set targets 1,704 loci. It spans a wide variety of gene functions, including those pertaining to immune responses, depth adaptation, reproduction, metabolic processes, cell death, and homeostasis. We will use our baits to study selection and other evolutionary processes in the genus Paramyricea across a depth gradient. Yet more generally, our bait set can be used by anyone wanting to study evolutionary processes in deep sea octocorals.
This project was made possible through an internship opportunity in the Coral Lab at the Smithsonian National Museum of Natural History. If you're interested, please check out our poster and reach out. Thanks for listening.
So Shriram and Ariane put this presentation together, and I thought it was a really nice overview. We have the data back. I'm not going to show it today. We are just beginning to dive into it. But in general, it worked fairly well. We were able to capture most of the loci that we targeted within Paramyricea and also several outgroups as well. So stay tuned for more results soon on this project. All right, so to wrap up today on the topic of the deep sea, I just wanted to highlight some of the deep sea exploration work that we've been conducting off of Puerto Rico. As I stated in the beginning of my talk today, characterization of deep sea biodiversity is extremely important and ongoing, and we absolutely need to increase our efforts to collect and sequence deep sea corals, particularly those in type localities as well. And the waters off Puerto Rico actually hold several deep sea type species as a result of expeditions in the late 19th and early 20th centuries. And one such expedition was actually led by the Smithsonian, by Dr. Paul Barsh at the Smithsonian Institution. This was the Johnson-Smithsonian expedition of 1933. And it was sponsored by Eldridge Johnson, who is a wealthy business owner from Philadelphia. And he just provided his yacht to the Smithsonian for use for an expedition to the Puerto Rico Trench and areas surrounding Puerto Rico. So in April 22, I led a cruise off of Puerto Rico again. There's been expeditions since this time to the deep sea off of Puerto Rico, but this was the first Smithsonian-led expedition. And we sailed in and out of San Juan, Puerto Rico, and we hit a few sites to the south of the island. So the pins that you know are the few sites that we hit. We had some issues related to COVID and also weather, and so we couldn't hit all the sites that we planned. But even in just these 4 places that had actually never been explored before, we really found some amazing discoveries that I'm just going to share with you today because I think they're super fun. We did use an ROV, the Global Explorer. We brought an intern from the University of Puerto Rico, Mayaguez, on board with us, Jose, and he came to the museum in just a couple months after that cruise and spent 8 weeks with us, along with another intern from the University of Puerto Rico, to actually work on the material that we collected during this expedition. We conducted outreach from the ship to shoreside with Eco Exploratorio, which is a museum and a mall in San Juan. And following this cruise, we conducted different outreach events at the Natural History Museum. One being an Expert is in event, which basically students and curators, myself, we all went out into the Ocean Hall of the Natural History Museum, set up stations, and kind of did a takeover outreach event with the public. You know, it was really important to me to make sure that our results were more accessible to a broader audience. So all of the content that we produce online was produced in both Spanish and English. And then during the Expert is In event, we made sure that there were bilingual speakers at every station that we had. We collected several really interesting invertebrates and new species, and I just wanted to show them off today. This is a new species of black coral. My postdoc Jeremy is really excited in describing this. It's about 1 meter tall and 3 meters wide, and it was collected around 400 meters depth. We collected a new species of soft coral seen here, growing on what looks to be a bag of cabbage. We saw cabbage written on the side of it, so hopefully that's just cabbage in there. Yeah, or it could be just a bag of rocks, maybe used as an anchor or something like that. We also collected this gold coral here on the right. I sent it to a colleague of mine in Australia, and she was really excited about it, as this likely represents the type for the genus Chrysogorgia and represents a type locality that it was collected from and described from. We also collected a new species of colonial tunicate, and this was a fun story because when we saw it, is at about, I think, 1,000 meters depth, we really weren't sure what it was. There was all bets that it could have been a sponge or a sea pen. And when we brought it on board, it was definitely a tunicate, and a really weird one. So we're looking for somebody to help describe this species. We also collected a new species of hydrozoan, which are also cnidarians. I don't know if you can see the tiny little whitish pinkish dots. This new species of hydrozoan, and it was growing as epifauna on a glass sponge. And the side note about this glass sponge, it provides a home to a pair of shrimp, a female and a male, and these shrimp are partners for life. This glass sponge has like a plate over top of it. It's called a sieve plate. And so there's no way to get in or out of this sponge, anything this big at least. So these shrimp are in this sponge, female and male, partners for life. And the last story to tell you is about this bryozoan that we collected. Full circle back, this species was described from a collection from the Johnson-Smithsonian expedition in 1933. And to our knowledge, it's not found in any other museum worldwide, except for the Smithsonian Museum. And it looks like it hasn't been collected since 1933 until our expedition this past April. We have sequenced its mitogenome. And we'll probably put out a small note about this bryozoan. The picture on the right, if you're curious, this picture is a close-up of its avicularium, which is basically a defense zoid. All right, so just to leave you with a couple big take-home messages from all of this work. Target capture genomics is really useful for resolving deep to shallow relationships in anthozoans. With this backbone phylogeny, we were able to determine that anthozoans originated before the Cambrian explosion. We were able to map the hallmarks of reef-building corals, the hallmark traits of reef-building corals, and determine when they originated in deep time. In general, coral diversity is grossly underestimated, yet it is really important habitat from shallow waters to the deep sea. And, you know, we really have a lot to learn about coral natural history, diversity, and persistence in the face of global ocean change. So with that, I just want to acknowledge some of my funders, particularly NOAA and NSF. And I want to just give a shout out to all of the undergraduates and interns who have helped with various aspects of these projects over the years, and want to kind of just all alert you to some opportunities at the Smithsonian. If you're familiar, or if you have students who might be interested, we have central fellowship programs through Smithsonian Institution Fellowship Program. Every summer we have a natural history research experience for undergraduates, and I believe this year will be open to international students as well. And I often have virtual internship and in-person internship opportunities. So if you ever have any questions about these programs, I'm happy to answer anything that I can, and please don't hesitate to reach out. And thanks again for listening and the invitation to talk today.
Well, I hope everybody's joining me in a virtual and physical applause for Andrea. That was really great. I guess we can open up the floor to questions. If you have a question, you're on the virtual space, you can just like raise your hand and we'll call on you. Or if you want to drop it in the chat, that works too. And I can kick it off since we don't have anybody yet. I'm really curious, Andrea. So like with corals, and stop me if I'm wrong, but like, you know, in a coral community, you have like hundreds or thousands of individuals living side by side. How do you deal with that when you do your genomic studies? Like, you know, do you treat it as these are all members of a population? Are they somehow, you know, relatives of one another? Things like that. That might be naive for me to ask that, but I'm very curious.
Oh, that's a great question. We often don't have enough individuals, especially the deep sea. I'm going to talk from a deep sea perspective, to look at, say, sibling relationships or anything like that. But we definitely screen for any clones when we can, population genetic approaches. When we do the collecting, I mean, these are colonies, and in all cases, we're just taking a small branch of each colony, and we don't really see a difference in different portions of that branch at all in terms of the genetics. That's what you're asking.
Totally. Yeah, that's really interesting. I had assumed that maybe there would be a lot of difference or not. It'd be either or. So that's cool to know.
Yeah, Zach.
Hey, can you hear me okay?
Yeah.
Okay, cool. So I was wondering about the mitochondrial mutation rate. Is there like any idea as to why the mutation rate in corals is so much lower than it is in other organisms?
Yes, sort of. In octocorals have a unique mismatch repair gene in their mitochondrial genomes. So that makes a lot of sense. They're actively fixing mutations. But hexacorals do not have this gene. So it's thought that there might be more nuclear mitochondrial connection where there's some nuclear genes that might fix the mitochondrial genome. They're also under very strong purifying selection. I mean, I know most mitogenomes are, but it seems like the omega values are quite low for the anthozoans in general. And the mismatch repair enzyme explains the octocorals, but not.
Right.
Yeah.
So one other thing, you showed a couple of like the commensals and stuff that you get whenever you bring up these corals. I'm curious, what do you generally do with them? Do you like contact other experts? Do they get deposited into the museum? What happens to all of like the other commensals and other organisms that are living on these corals?
Yeah, so from this latest cruise, everything just gets deposited in the museum. When we have visitors come through, visitors can access the collections, provide IDs. If they're interested in more than just identifying them, the material is available to them. But I will say, I mean, there's just generally a lack of deep sea taxonomists, and there's just so much, especially in vertebrates, there's just so many groups that we just don't have a good handle on yet. And, you know, the Okeanos cruises that Randy was talking about in the beginning of the introduction, all of those samples that are collected during those telepresence expeditions that are really driven by the public, they come to us at the Smithsonian. And to my knowledge, not a lot of people are requesting those. So they're available for others to use too. And they're all not only pickled, but put in the biorepository at the Smithsonian.
Thank you.
Yep. Yeah, Dan.
Yeah, I'm wondering what your expert opinion is on what the ultimate overall diversity by depth relationship will look like for corals in a general sense.
I think it's
in a final analysis, like subjectively interpreting what you know about cryptic species and sampling gaps and so on.
Yeah, I think it's very strong. I think a lot of the cryptic species are going to be seen within these different depth bands as we get deeper. I think the mesophotic depth area, so that's a little deeper than scuba. So I want to say 50 to 150, 200 meters. I think that area we're going to see an extreme amount of coral diversity, particularly of octocorals. And there's really just nobody really targeting that. Just a maybe one or two people that have been doing some targeted collections with tech diving.
we have a question from Yukai in the chat. It's surprised to learn that corals originated so deep in the Pterozoic, well, Proterozoic, sorry. Do we have any ideas about what they looked like and if we have support from the fossil record? Thanks.
Yeah, that's a great question. To my knowledge, no. The oldest confirmed anthozoan fossil was around 510 million years. I used it to date the tree, to put a prior on the tree. And I can't recall off the top of my head what that looked like. I'm pretty sure it was anemone-like, and it was found in about 510 million years. I had this question because I was a little worried too about the old origin of coloniality, and had a conversation with Doug Irwin, who's really thought about this a lot in deep time. And some of the work that he has done, he didn't think it was too out of the ordinary that we would see coloniality so deep in time.
Any other questions? Pressing questions about the deep ocean or octocorals or black corals?
Yeah, so black corals, although their tissue is red, they have proteinaceous skeletons that are black, and black corals are used to make jewelry in a lot of cases. There's some black corals that are actively managed for fisheries for jewelry, which is interesting, and also illegally traded all around the world too.
Dan's got another one.
I have another question. I'm just thinking about, the cost of biodiversity exploration in places that are hard to access. So I'm also wondering if you could give your informal assessment of, what is a typical expedition like this cost in terms of, for typical duration that people go out for, and, focusing on even a relatively close area like, off Puerto Rico, for instance. And sort of what do you think it needs in terms of cost if you were going to get a pretty good first order approximation of, let's say, global patterns of biodiversity in the deep sea for corals, just even in terms of just getting the actual material?
No, that's an excellent question. So that cruise that I had was 2 weeks. We got free ship time through a NOAA scientist. We got free NOAA ship time, and the ROV cost $350,000, and that is like not the top of the ROV line. If you wanted to use something like the Jason ROV or the Alvin submersible, I mean, you're looking at like $60,000 to $75,000 a day just for the vehicle, and then you add another $50K sometimes on top for the ship. So $100K a day, times 14 for a 2-week cruise. It's really expensive, and this is really limiting our ability to access places that we know even less about, like the Indian Ocean, for example, or off the coast of Africa in the Atlantic, just as an example. I think there's a push in the deep sea community to make more open access resources, to utilize like smaller open access ROVs and things like that. But I mean, I really do think you need some of these bigger ROVs that can work a little bit better and collect more to kind of get at some of these larger biodiversity questions. I think we're going to also see an uptick in eDNA use in the deep sea. It's a lot easier and cheaper to take a ship with, say, a CTD, so a connectivity temperature depth recorder that you can trigger bottles at certain depths and take water. And I think we're going to see an increase in that. And I think that's okay as long as we build up the reference database for eDNA work. But I think there'll be definitely more eDNA in the deep sea in the upcoming years. It's a lot cheaper.
I definitely agree with that collaborative effort. Like, I feel like sharing specimens and data is like the bread and butter of our work.
Yeah, and as a person who works at a museum too, I think our museum collections, and for all of you that work at the museum too, that they're just really a treasure trove of things that we just haven't even really completely gathered all the information we can from museum specimens. And so that's one effort that I'm pushing at the Smithsonian, is to try to dig in to what we have. We're able to get DNA out of dried specimens really easily and really try to increase the, you know, the genomic data and all the stuff that we already have collected in the museum.
All right, well, on that inspirational note, I think we'll wrap things up. But you know, it is unfortunate we aren't able to like take you out and socialize and everything tonight. But you know, hopefully we can get you back here and you can meet with people again and all that. So we'll find an excuse to get you back.
Thanks so much, Randy, for the invitation. I appreciate it.
Yeah,
Fun.
thank you, everybody.